This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 900, recorded on May 12th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello there, Vincent. I'm just, when I hear you say Microbe TV, I immediately try and boot up the Weather Channel to find out what's going on. And for some reason, it's not showing me Austin. At any rate, I can tell you, it's probably about 80, 85 and sunny. Not a bad day. Though it's been really hot recently, and we're looking at 100-degree days well in advance of what they ought to be. But that's good. I went rowing this morning. So
nice.
It was nice out. Yeah.
It's 21°C here in New York. I was in Omaha yesterday. It was 92. Wow. They said it was unseasonably hot.
Yeah.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is 81 Fahrenheit, 27 C here in Western Massachusetts. And the past few days have just been gorgeous early summer weather. I've been mulching flower beds in the mornings, which is an ongoing chore. And yeah, it's good. So I made a mistake. I would usually at these every 100 TWiVs, I introduce people in the order that they came. And Alan should be first and then followed by Rich. Also joining us today from Ann Arbor, Kathy Spindler.
Hi, everybody. And I made a mistake by reacting to Rich rowing and
okay.
spoke before I was introduced.
No worries.
It's because I've been rowing in that same place with Rich, so that was fun. Here in Ann Arbor, it's 85 Fahrenheit, 28 Celsius, and wispy white clouds, but really nice day.
And from Madison, New Jersey, Brian Barker.
Hi. It's great to see everyone. It is 73 Fahrenheit. 23 Celsius and sunny and just lovely here. And I turned in my grades yesterday, so I'm done for the semester. So that makes it even lovelier.
No. Well, my exam is not over till 4 p.m., my final exam, and then grades are due supposedly by close of business tomorrow, but I'll get them in Saturday. I have like 90 extra credits to grade too. Maybe I got to get my TAs to do that. It's all right. It'll get done. Anyway, 900 is a cool number. We were at, I don't know, somewhere over 500 at the beginning of COVID. And man, we went through 600 and 700 and 800 and now 900.
And we should probably comment, Dixon was unable to make it.
Dixon and I started TWiV. We were the two who started it. Number one, which was in September 2008. And then Alan joined us on episode 10.
Yeah.
Which was called Bats, Elephants, and AIDS. And then Rich joined us on 41. I think these numbers are right because I had to basically go back manually and look for them. There was no other way I could figure out. And that was, 41 was fish flu. And then Kathy joined on 196, an arena for snakes. I think that's right.
Yep.
And we had a guest, right? That we had.
We did. We had Joe DeRisi and
Mark.
Yes,
Mark Stenglein. Yeah. It's funny. Mark was going to come on, and then he said Joe wanted to come on.
And you had been trying to get Joe, so it was kind of a coup.
It was very good
the way it came about.
And then Brienne joined us in 504, not too long ago, and. These are fun episodes because it's a nice number, and so we celebrate them. We've done different things over the years. Let's see if I can remember. 100, we had David Baltimore, right?
Mm-hmm.
Yeah, that was a big deal, 100.
Yeah.
And he came. He was at Columbia, so he came in my office, and that's back when we were doing Skype, so you guys just listened in.
Yep.
Then 200, I don't remember. Did we go to ASM?
Threading the needle?
That was the needle.
Yeah.
Okay.
Yeah.
300 was ASM, right?
Yes.
Yeah. We all went down to Washington. 400 was Harold Varmus.
There at Columbia.
Yep.
Yes, we all came to New York
Harold
for
400
that. Right?
Varmus. Yep.
And then we went to dinner that night, right? If I remember.
Mm-hmm.
Yeah.
We did.
500 was in Austin.
Yep.
And then 6, 7, and 800 were on Zoom.
Yes.
They're a blur.
Yes.
Well, you should know that Vincent said at 500, we're not going to do anything again until 1,000.
Yes.
And I think he thought that was going to be way off in the future.
Yeah.
Well,
when he
we're not
said
really
it.
doing anything, right? But
Yeah,
we're
we're
just
not.
celebrating. But we're
We
not.
did make an extra effort to get everybody together.
Yeah,
We did,
I did
yes.
try and do that. And I hope for 1,000, all of you can come here
Yes.
to the incubator. And I will try and get the date far enough in advance. Right now, I'm not sure how many episodes a week we're going to be doing, I would like to cut down to 2 around June 1st. So that would be Daniel's clinical update and then a TWiV.
Yeah.
And I don't know how long Daniel wants to do his. I don't know if you guys know this, but Daniel and Sarah Dong have started a new podcast, the Infectious Disease Puscast,
Yeah, I saw that online.
taking over for Mark Crislip's
Right.
podcast. He stopped his. And we asked him if we could use the name, and he said it was fine. So that's twice a month. So he might want to cut back on the clinical updates. We'll see. But I suspect we will not do 1,000 this year, maybe next year, or maybe not. Who knows? But anyway, it's a lot of fun. And I want to thank all of you for doing it, man. It's
Yeah.
great.
Well, thank
You
you.
were,
Yeah,
doing,
thank
you
you.
know,
Yeah.
we're
thank
all
you.
doing this together, and I appreciate you guys make time. And people really like it. That's what drives
Yeah.
me, is that people love it. I was just in, as I said, Omaha. I was invited by the grad students, and they all love TWiV, right? They just listen to it, and they think that that's how they learn. And in fact, I talked to a young faculty member who was a graduate student at Mount Sinai with Kathy, the guy who went to Atlanta. Remember his name? The Ebola guy. I can't remember his name. He was on TWiV also. TWiV, Ebola. This is not a good sign. Atlanta.
Somehow those
900
cues aren't-
episodes, you start to lose track of exactly what was when.
What is it? That was Georgia State. Yeah. Georgia State. Let's see if that helps. Yeah,
And it's
there
been
it is.
14 years, right? So we're coming up on 14 years.
Yeah, that's true. Chris Basler.
Oh, right. Okay. Sorry.
Anyway, so he was a grad student with Chris Basler, and he said, so now he's a faculty member at Omaha. He said, all the students listen to TWiV. He said, we all learned from it because it's a great way to listen to new papers in science. He said, yeah. He said, you have no idea how many people, students listen. So that's great. I really think that's what drives us, I think.
Well,
I'm
Vincent-
just waiting for you to say, like, hey, I met Caleb at a meeting. He's got his own lab now.
Yeah.
Yeah. I just wanted to say thank you, Vincent, because this is so much fun.
Yes.
It is a lot of fun for the most part. Yeah. Yeah.
Well, I think about, without this, I think that I would have pretty quickly after retirement just faded away into something else. And
That'd be a shame.
this has kept me in the game,
That's good.
and I love it.
Yeah. I think it's great, and we have a lot of fun. So we're gonna keep on going as long as we can.
Yeah.
So today we'll do a paper, but I had a snippet which I thought would be fun to do, and that is, I was asked to go to Zurich. I'm going Saturday to get this Richard Ernst lecture, to do a Richard Ernst lecture, and I'm going to get a gold medal. And so I hadn't heard of this, but it's given by Department of, I think, Physical Chemistry of ETH Zurich. So it's quite interesting because, as my host wrote to me, the aim of the lecture is to strengthen the relationship and understanding between the sciences, society, and politics, and to raise awareness for the questions and challenges our global society is facing today and will face in the future. Notice they put politics in there. The lecture is a public lecture for a scientifically interested general audience. So Richard Ernst, who was in that department, was the 1991 chemistry Nobel laureate. And apparently he was very interested in discussing how scientists' work interfaces with the public, right? Not communication per se, but knowing how what you do impacts people. And so, they wrote the ongoing COVID-19 pandemic and the accompanying public discourse have shown more than ever how important the education of the general public about scientific topics and the process of research is. With your series of podcasts where current research in virology, immunology, microbiology, et cetera, is discussed with experts in an accessible manner, you provide an invaluable service to the general public and the scientific community. We very much hope you'll be able to accept our invitation. I was invited, I think, in 2020. It was supposed to be 2021, and they canceled it multiple times, but now I'm going.
Excellent. That sounds like a good time.
And this is an amazing list of people.
This is quite a list of past recipients.
This is just ridiculous. I should not be going here, but the previous recipients, Emmanuel Charpentier, who I know, Stephen Chu, Roger Penrose, Kofi Annan.
Kofi Annan, yes.
Former UN Secretary General.
Yes.
Oh, my gosh. Stephen Chu, of course, was physics Nobel laureate and the energy secretary in the Obama administration. So there's quite a bunch of very accomplished people. And so I don't know. I'm very grateful that they think what we have done is important. So I'm going to go give a talk on, I think it's Tuesday. And I'm going to talk about, so this idea of interfacing with the public, there's two aspects. There is thinking about what your work how that impacts the public. And actually, we've done some things that fit into that category. We made the first infectious DNA of an animal virus, and from that stems all the debate about gain of function. So I can tie into that a little bit, right?
Yeah.
And what I'm going to emphasize is that when we did that, we didn't even think about gain of function. We knew we were going to do it, but we had no idea about this public discourse that ensued, really starting with the avian influenza business, right? And I think also making a transgenic mouse model for polio with a receptor gene is an example of gain of function with a mouse, right?
Sure.
And that from both of those discoveries now, a lot of work is done on SARS-CoV-2. And I want to emphasize that sometimes you don't know where your work is headed, and sometimes it has an impact that appears many years later. And then, of course, the other part of this is actually communicating, which is what we do. And I'm going to talk a lot about what it is that we do, what motivates us, who's listening, and just go through all the things, the blogs, the podcasts, the online lectures, and more recently live streaming, which is very interesting technology. So I thought that would be interesting for a science-interested audience.
Yeah.
Well, that's what they're getting anyway.
So, Vincent, I've been looking up Ernst Ludwig Winokur.
Yeah.
Because that name is so much like the name of an adenovirologist.
Yes.
Is Ernst L. Winokur.
Winokur, yeah.
So I don't know if it's the same one or not, because his bio calls him a biochemist and a geneticist and doesn't mention virology. But how could there be two Ernst L. Winokurs? I don't know.
It's interesting. That's a good point, yeah. I don't know. I don't know this one for sure.
I'm thinking of a Winokur that's spelled differently, who I think of as a polyoma virologist or something like that.
Well,
it was in Israel.
This is definitely E.L. Winokur adenovirus DNA spelled exactly the same way. And then when I click through to the paper, his name is Ernst L. So anyway, I don't know.
Anyway, there's a website for this, and I'll put that in the notes. You can check it out. And I'm looking forward. It should be fun. There's only one issue that I'm going to tell you about. So there's a talk begins at 4 p.m. There's some presentation, and then I give a lecture, which I've called An Inordinate Fondness for Viruses, which was my ASV talk also, because I think kind of fits for the theme they want. And then we're going to do a little panel discussion afterwards. They asked me to do this. And there are a variety of scientists there. And then one of the participants is Katrin Zoffel, who is a science editor at Swiss Public Radio. Should be interesting. And then they're going to have a musical interlude at the end. The Physical Chemistry Horn Ensemble.
Yes.
That should be great. Should be a lot of fun. And so the issue is that in order to get back in the U.S., I have to test negative for SARS-CoV-2. So I'm going to go on Monday morning before I go to the campus, and my host has figured out where I can go and get a quick turnaround PCR test. And
does it have to be a PCR test for the U.S.?
That can actually be antigen, but she just found a
Are you
PCR
taking
test.
all your podcast stuff in case you get stuck there?
Well, the issue is that on Thursday we have the official opening of the incubator.
Oh, dear.
Right?
Oh.
And so it will have to proceed without me because we have some people coming from pretty far away.
Right.
And so, Daniel will be here. Dixon was going to go. I'm not sure he will. Several of the other podcasts, Rich is coming. Cindy Leifer, Brianne is coming. And some scientists from around New York City and so forth. Steve Morris
and you're
is
going to
coming.
leave the keys under the mat so people can get in, right?
Yeah, well, Daniel has keys.
Daniel has keys.
But I told him, just continue with it in my absence. I'm going to tell him, I have a fundraiser who is really taking care of it. He's got catering and so forth. And
well, I hope you're able to make it.
Yeah, making all those contingency plans, and then you won't need them. So
hopefully
it's
not.
better to be prepared. Yeah.
I have been masking for the past week on all the public transport, on the flight to and from Omaha. I'm going to mask on the flight there. If I figure I just don't want to be exposed, right, and have a PCR test. I'm not worried about getting sick. I just want to be back here. So it's funny, isn't it, that this is what I have to deal with. But
Kathy, go ahead.
I asked Daniel, I said, hey, could I take prophylactic Paxlovid for a week? He said, no, you can't. I even thought about getting another booster. Oh my God, horrors. But I didn't do it. And it's too late now. Anyway, so that's what we're doing. Yes, Brianne, sorry.
I was going to ask if Kathy knows what E.L. Winokur looks like. Because the one listed here does have a Wikipedia page, and it says his main fields of research are virus cell interaction, the mechanisms of gene expression in higher cells, and prion diseases.
Okay, then it seems like that was him. I did see a set of images also, but it didn't quite look like what I remembered. But people age and change, and I might not have known all that well who he was in my younger days. So yeah, it seems like too coincidental to not be the same person.
So by the way, ETH is a public university in Zurich. Does anyone know what ETH stands for?
Oh, I looked this up for something and I've forgotten it. It's something
I was going to ask. I
in
do not
German, know. I think it's. Yeah.
Let's see. Yeah, it must be German. ETH Zurich. Science and Technology. Well, you could just say what does.
So I can see the German, though I cannot pronounce it.
Too.
But it's Swiss Federal Institute of Technology in Zurich.
Got it.
Eidgenössische Technische Hochschule Zurich. Yeah.
Got it. There you go.
Sorry, it wasn't the greatest either, but close.
Okay. Yes, I think I should know before I go, right?
Yeah.
Okay, so that's our snippet for today. And we also have a paper for your education. And this is a paper came across a couple of weeks ago, which I think is quite important. It is a preprint or accelerated article preview in Nature.
Right. Yeah, not a preprint. It has been.
It's been reviewed.
But they're just letting you know, don't add them about typos. It's
yeah.
Oh my goodness.
There are lots, yeah.
Well, It's not so much typos as there's a place where they need to buy a verb.
Yes.
And then there's another place where there's two sentences in a row that have two verbs and they need to buy conjunction.
Yes.
And and numerous other things. It's just very figure dense.
Yes.
Oh, yes.
I mean, so I'll just tell the listeners, there's 4 figures. I think that's all you're allowed for nature.
Mm-hmm.
Each of the figures has parts minimum A through I.
Yes.
And some of those parts have multiple pieces
Yes.
of data in them. So that's the 4. So let's just say there's 20 to 25 pieces of data in the main 4 figures a piece. So that's 100 pieces of data. And then there are 15 extended figures, each of which has on the order of 20 more figures or parts
Yeah.
or whatever. So you're talking 300. It just boggles the mind.
It is a lot of work.
And nature restricts the length of the text as well. So we get a 1-paragraph introduction and then results. So it's
Yeah, and this is one of these things where, I mean, you know, the way I was sort of brought up and the model I followed was that, you know, you actually described what the experiment was in the text, and then you describe the results and stuff. And what this does is it's, you know, and it's not necessarily their fault. There's a lot of data and restricted space, but this is each paragraph is a sequence of statements of results.
Yes.
And then you go look at the figure, and you got to, from the figure, try and kind of figure out what the experiment was. So to tell you the honest truth, I kind of zoned out at one point, and when I was done with the paper, in my mind, I summarized what the major findings were,
right,
and went back and looked at the figures and looked at the titles of the figures, because the titles of the figures basically are the 4 major observations in the paper, with a slug of data supporting each statement that's the title of the figure. And if I kind of took a 30,000 foot look at it from that point of view, I'm okay.
Yeah.
Okay. But getting down into the weeds on this one is tough.
Yeah.
But it's good, because these are broad conclusions.
Yes.
Of significance. Okay. And so it's good to have 100 pieces of data for each one.
Yes.
Big claims require a lot of support, and the support is in there as far as I can tell. But like Rich, I kind of went through okay, wait, what are we getting to here? All right, we're getting to this. So I'm
maybe it would be good to have Rich read those 4 figure legends, or how you restate those 4 pieces of data as sort of a, broad overview of where we're going first.
Sure. That's a good idea. Let me see. Let me pan down here and find those.
And while you're looking, let me tell you, this is from quite a few authors at Yale University, Harvard Medical School, and Fundação Oswaldo Cruz in Brazil, the Rockefeller University. First author is Isin Sifik, and included in the paper are Kiko Iwasaki, Michelle Nussenzweig, Judy Lieberman, Craig Weiland, who I think we know on TWiV.
Mm-hmm.
Didn't he get a technician through TWiV or something? I just don't remember the connection.
I thought he was on TWiV.
You don't remember?
Or we did one of his papers at least.
Yeah. Anyway, and then the last author is Richard Flavell. Okay, Rich.
And I want to embellish on that and say that I have a connection with Richard Flavell.
Uh-huh.
I did a postdoc with his younger brother. I mean, alongside his younger brother. He and I were postdocs together in Bob Kamen's lab. And also Bob Kamen was a postdoc together with Dick Flavell in Charlie Weissman's laboratory in Zurich. And at the time, Dick was on the faculty at the University of Amsterdam. And when my wife and I took a walkabout after being in London, Dick quite generously, I don't think he was even home, basically gave us the keys to his place, and we crashed in his place in Amsterdam for a couple of days. It was great because there was a canal out the back. Okay, you couldn't see the canal, but you look out his back window, and it would look like you're looking across a field, and this big old ship is sailing across the field. At any rate, good on you, Dick.
Yeah.
Okay, figure 1 is targeting viral replication and downstream interferon signaling ameliorates chronic COVID-19.
Okay.
So this is basically saying that chronic COVID-19, as revealed by mostly this mouse model that we will discuss, is a function of a combination of virus replication and the immune response.
Right.
Okay. And that if you tinker with either the virus replication itself or the immune response, you can impact the chronic long-term infection. Figure 2 is SARS-CoV-2 replicates in human macrophages. Okay, so this is an important part of the whole thing because macrophages are important in inflammation and the immune response. And there's a whole slug of data supporting that. Figure 3, SARS-CoV-2 infection of human macrophages activates inflammasomes and pyroptosis. Okay, so the macrophages blow up. Okay, they go nuts and eventually die.
Yeah.
Okay, not before sending off a lot of alarms.
And we'll,
Is that okay with
I'm
you,
sure
Brianne?
That is okay on me. If you would like to hear more about the inflammasome or pyroptosis,
I'm sure we'll get there.
I think we need to. We'll get there.
And figure 4 is figure 4 is inflammasome inhibition ameliorates inflammation and disease in infected humanized mice. Okay, so the idea is this inflammation. The macrophage activation and pyroptosis and the activation of the inflammasome is not just a sideshow, that if you inhibit that, you can actually impact the profile of the disease. So those are the basic things. There's interferon and virus replication involved in chronic COVID. The virus infects macrophages. That activates inflammasomes. And results in pyroptosis, which is a specific kind of death of macrophages, sending off a lot of signals in the process. And that if you interfere with that inflammatory response of the macrophages, it impacts the profile of the disease.
Yes.
Fair enough?
Good. Yeah.
Good.
All right. We can stop there. Yeah.
This 2 other things. This paper is open access, so everybody can go download the accelerated preview. And when you do so, you should definitely look at online because there are 15 extended data figures that go with it, each of which also goes, most of those go to I in the subpanels.
So this is a good paper for 900 because I think it illustrates what the kind of paper we want out of COVID, right?
Yes.
Where you get mechanism and understand what is going on, not just observations that you don't understand. I mean, it's hard to do. There are a lot of people on this paper with different skill sets and a lot, as we have said, a lot of experiments. But I thought this is going to be an important one. And, in the end, they say we could probably make some therapeutics now based on what we have found for severe COVID.
Yeah, actually, the mechanism thing is key to this whole thing, because if you look back at the first figure, we'll get to this, one of their controls for looking at the inflammation is dexamethasone.
Yes.
Okay, which is one of the most effective in these experiments inhibitors of the inflammatory response. And we've known since early on in the pandemic, empirically, that treating people with dexamethasone is a good thing to help prevent severe COVID. Now we're touching on mechanism for how that works.
Yeah. And I think that one other thing that people who are really interested in this should be aware of, this may be me geeking out a bit, is that if you look at this paper, you will note that it was submitted a long time before it was eventually published. So there's a big gap there where it was submitted in July 2021 and published in April 2022. And there is actually a somewhat similar paper that Vincent and Amy talked about on TWiV 887. And it also, I remember, it's from some of the same authors on this paper, particularly Judy Lieberman and Caroline Junqueira. And it also took a while in review. And so what I'll say is that these are big claims that are really interesting claims, and big claims require a lot of evidence behind them. And so that's part of the reason why there are all of these giant figures that we've been talking about, is because this is a big deal.
Yes. And this is mechanistic insight, not just on an aspect of the pandemic, but what I would point to as the critical question in the pandemic. Why do some people get so sick with this virus?
Yeah.
Because
Exactly.
if that wasn't the case, this would just be another common cold and nobody would care. This is the core of the problem.
Exactly right. Now, there's an introduction of one paragraph.
Yes.
And it depends on you going to other papers to substantiate what they say. For example, they say acute SARS-CoV-2 infection resolves in most patients, but in 10 to 20% becomes chronic and sometimes deadly. And then there are 1 to 7, a ton of references for that you have to go. 2 hallmarks of severe, so this is severe COVID, sustained interferon response and viral RNA persisting for months. And again, multiple references. That pattern, what they call a chronicity, is recapitulated in humanized mice. And these mice are MISTRG6 human ACE2 humanized mice. And there's a single reference for that. And then they describe how they make them in the methods. And we'll get to that in a moment when we do the results.
Do they call them Mr. G6 mice?
I would call them Mr. G6.
That's good, Mr. G6.
Yes.
Yeah. Mr. G. I like that. Do you know, Brian, do they call him Mr. G6 mice?
I have never heard of them. I don't know if they're called Mr. G6, but I think that sounds really good.
Yeah.
That's good. I like it.
It'd be a good nickname for a pilot of private jets.
Yeah, exactly.
Now, in patients with severe COVID, what correlates with the severity is what they call copious IL-1 beta, IL-18, and lactate dehydrogenase, LDH. And they say this suggests a role for inflammasome activation and pyroptosis in pathology. And Brianne, so those are hallmarks of inflammasome activation, correct?
So both IL-1 beta and IL-18 require the inflammasome in order to be processed and to be secreted. And lactate dehydrogenase is an enzyme that's normally in the cell that can leak out of the cell when the cell undergoes cell death by pyroptosis. I'm sure I'll make lots of comparisons between pyroptosis and apoptosis, but here, in this case, it's sort of important that you think about pyroptosis as maybe being slightly less organized than apoptosis in terms of all sorts of enzymes like LDH leaking.
All right.
And it's unique to pyroptosis. In other words, you don't see LDH leakage in apoptosis.
Right, exactly.
Okay.
And the inflammasome is this structure that it's kind of like the inflammatory grenade, I think of it as. It's right.
Yeah, it's a protein complex that involves a protease that will cleave a number of substrates to lead to the cell's death. And it's a particular unique kind of death, pyroptosis. One of my colleagues likes to call it the pinwheel of death because it does look like a pinwheel.
So the inflammasome is activated by sensing of certain PAMPs, right? Mm-hmm. What is sensed to activate the inflammasome? We don't know.
That's a hard question. There are a few different sensors. Some of them we understand the PAMPs. So for example, there's one called NLRC4. And we know that things like flagellin in a type 3 secretion system can turn on NLRC4. However, there's another one called NLRP3, and all sorts of different things turn on NLRP3.
Okay.
And so that one makes it a little bit harder to tell.
All right. So they want to know if indeed the inflammasome is involved in severe COVID, and they're using a mouse model, but they do have some human data sprinkled about, which illustrates nice ways to correlate with mouse data. So the mouse model, Mr. G6, ACE2 mice, I thought we would just tell you a little bit about that. So you start with immunocompromised mice, RAG2 null mice, IL-2 common gamma chain null mice, right? Which what? They don't make any TB or NK cells, Brianne? Is that correct?
Yes. So no T cells, B cells, NK cells.
God, I've learned something in the past 2 years. And then they put into that background human genes for MCSF, IL-3, SRP alpha, thrombopoietin, GM-CSF, and IL-6. They knock them into the mouse loci. So they replace the mouse counterparts, right? That's correct.
That's what that means, yes.
So they took out a big chunk of the mouse immune system and put in the equivalent chunk of the human immune system.
Well, no, here they have not yet put in. Right. They have taken out mouse B cells and T cells. So basically the mouse adaptive immune system is gone.
Right.
They've put in some human immune genes. And then there's another part where they actually also put in some human cells.
Right.
Yeah. So then they take human fetal liver pieces and purify stem cells from them and inject them into newborn mice, basically to engraft those cells into them, right? And so that's why you have immunocompromised mice, because you don't want these cells to be destroyed, right?
Exactly.
And then finally, if you want human ACE2 in, then you deliver that by an adeno-associated virus vector, and you inject the virus into the trachea. You actually open up the chest of the mice. You see the trachea sitting there. You put a needle in it, and they put 10 to the 11 genomic copies of an AAV carrying human ACE2 gene.
So, Brianne, is during development in a fetus, is the liver the major or a major site of hematopoiesis?
Yes. Fetal liver is the major site of hematopoiesis in early development.
And that gets turned over to bone marrow eventually?
That does. It gets turned over to bone marrow, and I am picturing the slide I have of that, and I cannot tell you what the axis is in terms of timing, but it does eventually turn over to bone marrow.
So with knowing almost nothing, my assumption was that the knock-in of the human genes were kind of made the mouse hospitable for this transplantation of the human immune cells. Is that a fair statement?
I guess so. I am actually more familiar with this being done in just the RAG2 deficient, gamma chain deficient mouse. So the Mr. G6 is unique to having these additional genes in as well. In fact, if you look at the genes that are overexpressed, or the human genes that are expressed you look at the first letters, you get Mr. G6. I am less familiar with those, and am more familiar with just the what are called humanized mice that have RAG2 and gamma chain deficient plus the fetal liver cells.
So in this mouse, there's no mouse immunity at all. Is that right?
There should be a little bit of mouse innate immunity, but most of the really critical innate immune genes have been replaced by the human versions.
Importantly, it's not going to make mouse macrophages, right?
It might make mouse. No, it does make some mouse macrophages.
Yeah, actually they do, because that comes up later in the story.
Yeah. No, it makes mouse mouse. Yeah.
So it is Mr. G and Alan. There's got to be some title. Mr. G goes to somewhere.
Mr. G goes to
goes to COVID. I don't know. You can work on it.
Yes.
So they say now in these mice, Mr. G with human ACE2, this chronicity of severe COVID is recapitulated, and that's all previous data that they've been published. So now they want to know what is the driver of severe disease in this mouse model. So they say to test whether an RNA-dependent type 1 interferon response drives chronic disease, they infect these mice with SARS-CoV-2, and they treat them either with remdesivir, which of course inhibits viral RNA synthesis, or an antibody to the type 1 interferon receptor, right? So you're inhibiting virus replication or the interferon signaling, the interferon binding to its receptor. And their control, as Rich indicated, is dexamethasone, which they've previously shown that reverses much of the immunopathology in Mr. G human ACE2 mice. I'm just picturing him wearing a nice cardigan.
Yes.
So they say alone remdesivir and anti-IFNAR antibodies had some effect, but together the mice recovered their weight loss and the inflammatory response was suppressed. As good as dexamethasone, which of course is used to treat patients, as Daniel has said many times, as long as you give it at the right time. Not early. Not early, because you'll impair virus clearance. And so that's the first bit of data. And then, of course, they next look at the lung transcriptome. What does remdesivir and anti-IFNAR antibody do to the lung transcriptome? An obligatory experiment these days, right? Which generates tons of data that you need bioinformatic people to go through. And they say that they know which transcripts go up in the lung of this mouse model. And they both dexamethasone and the combined therapy reverses what they call overactive immune transcripts to uninfected animal levels, and these encode transcripts in chemokines and cytokines, inflammatory proteins, antiviral, ISGs, and so forth. All of those go up in these mice when they're infected, and the treatment brings them back down again. They also have single cell transcriptomes of human immune cells from these mice, because remember, they have some human immune cells in them compared to uninfected cells. And this shows them that the tissue-resident macrophages, like alveolar macrophages, are activated at the peak of infection. Then you get an inflammatory response, and monocytes then infiltrate into the tissue and they maintain this inflammatory signature throughout infection, making all of these transcripts at high levels. All of these, and then ISGs are part of this signature of enhanced transcripts, and they're all suppressed by the combination antibody remdesivir therapy. But they say even with those two, some key antiviral responses like interferon gamma, which is made mostly by CTLs, is not affected. Now, I was a little bit confused with this. So apparently part of the clinical picture in people and also in the humanized mouse model is fibrosis. They write, consistent with fibrosis in patients in humanized mice, alveolar self-renewal and differentiation programs were inhibited. That's by the combined therapy, right? Inhibited by the combined therapy, resulting in the accumulation of pre-alveolar type 1 cell types. Oh, no, that was reversed. So that must be infection.
Right. So infection stops the types of self-renewal that you should have in the lung. Yeah, as a result, the lung is damaged.
That's the fibrosis, right?
Yeah, fibrosis is the damage. And when you give the treatment, you stop that abnormal differentiation. And so now the lung can repair itself normally.
Okay. So can you elaborate on fibrosis? Does that involve an infiltration of I'm blanking on the words here.
I can't elaborate on it too much, although it's sort of an area that I've been trying to read about a little bit more. But there are definitely linkages between immune cells, particularly macrophages, and the ability to make things like clotting factors and other types of kind of extracellular matrix, both of which I think are involved in fibrosis.
Okay.
So the conclusion is that reducing chronic inflammation with this double therapy enhances the recovery of the lung tissue and prevents fibrosis, which you would normally see after infection, both in humanized mice and in humans. Okay.
Yeah. And now, at this point, when I look at data like this, I'm sort of intrigued because everything they've seen so far sort of says, look at all of these things that are happening that are related to the pathology that also happen to be related to monocytes and macrophages. And so you could imagine a few different things going on. One of them being that the virus is infecting the macrophages. The other being that some other aspect of infection, maybe debris from infected cells is somehow activating the macrophages. So we've sort of implicated there's a problematic macrophage response, but whether that's because the macrophages themselves are infected or whether that's because they are sensing something else happening because of infection is not clear.
Right.
This is the point where I think from bits and pieces I've heard through Grapevine that this is what took a while with some of this work to get published, was making everyone feel comfortable about whether or not the macrophages were actually infected.
Thank you, Brianne. That's a great setup because, I don't know enough to ask those questions. Okay. But clearly you're ahead of the game here.
Well, they don't, the brevity of the manuscript prevents them from setting you up properly, right?
I heard one of these authors give a talk before these papers were published where they clearly had something cool that they wanted to talk about but couldn't talk about. And it was kind of, you could kind of tell it was about whether or not macrophages were infected, but they couldn't officially tell you that.
All right, so the next section addresses this question: is the virus actually reproducing in macrophages? So they make a virus which carries a gene encoding a fluorescent protein, and they can use that to assess infection of cells. So when they look at fluorescence, they say most epithelial cells in the bronchoalveolar lavage, but only few total lung epithelial cells were infected. Now, Brianne, a bowel, what's mainly in a bowel? Are they immune cells mainly, or? I
think there should be some immune cells, yes.
So it's a mix of epithelial cells that come off and immune cells.
And immune cells that have infiltrated.
All right.
The epithelial cells are going to be from the mouse.
Right. In this case, they're mouse.
And they should have ACE2, right? Because they put the AAV in. They put the AAV vector in, so they should have ACE2 and be infected.
I believe they should be infectable.
Yeah, infectable. They say, strikingly, human macrophages were strongly fluorescent throughout disease. No mouse immune cells made this fluorescent protein, virus protein, so they're apparently not infected. All right, so it looks like the human cells, in particular macrophages. Now, they're strongly fluorescent, and that's interesting. So the virus is clearly getting in. It's making mRNAs that are translated to form the fluorescent protein.
Right, but then if you're a super picky immunologist, you say, well, macrophages are highly phagocytic cells.
That's right.
Are the macrophages actually infected? Have we, seen attachment to a receptor and actual parts of the infectious cycle, or did the macrophages just phagocytose?
Right. So they asked whether the virus is actually reproducing. They say, they stain for double-stranded RNA, which they say is diagnostic of viral replication, and they can find fluorescent signal and double-stranded RNA in human macrophages. They can find the viral RNA polymerase in human macrophages along with the spike protein. And they say if you look in human macrophages from human autopsy lungs from people who died of COVID pneumonia, you can also find the RNA polymerase and spike protein. So that's this section. That's all they do. And they say we have similar results in the mice and in humans. We see the macrophages have these features, double-stranded RNA, RNA polymerase, and spike protein. And they fluoresce. In the mouse model, we see fluorescence.
So I know I sounded harsh on these folks at the beginning for the writing and stuff, but this is a place where, at least where they are talking about the a co-localization of the double-stranded RNA and the fluorescence. They refer you to figure 2C, and then they say also extended data figure 7A. And you really only see the co-localization in the extended data figure. So I know they had to make choices as to, they had to make hundreds of choices as to which figures to put in the main part of the paper. But that one I would have liked to have had in the main part of the paper and not have to go digging through the extended figures.
Yeah. It occurred to me that one of the sources of delay besides reviewers was just getting this many people to agree on what to cram in.
Yeah.
Mm-hmm.
All right. Now, how, as Brian said, is this actually getting in by a viral entry pathway, or is it being phagocytosed? So they look at that in the next section. So they treat Mr. G6 mice with an antibody against ACE2. And now in these mice, there's no AAV in these mice, just Mr. G6 alone, no AAV in those. In those mice, the virus infects the epithelium poorly. There's no human ACE2, right? In the mouse, ACE2 doesn't work well with the virus that they're using. Now, in now these mice have human cells in them. They have human macrophages, right, from the engraftment.
Yes.
And they have ACE2. And the ACE2 blockade blocks infection of those human macrophages by the criteria, the fluorescence and double-stranded RNA and all of that. So suggesting that the entry of the virus into these human macrophages is mediated by ACE2.
Or can be mediated by ACE2.
Can be mediated
Yes.
by ACE2. And then they said, well, antibodies can get virus into macrophages, right? We know that. And they say, for example, dengue virus. So they say, let's test the role of antibody-mediated viral entry at the macrophage. So the idea would be the antibody binds the virus, the FC portion of the antibody binds the FC receptor on the macrophage, and then it gets into the macrophages that way. So they have infected mice. They treat them early in infection, 35 hours post-infection, with an antibody against the spike. And they say these treatments indeed increased the infection of lung macrophages. So suggesting that the antibodies are mediating uptake. So what's the FC receptor that's involved here? And in fact, they say that severity of COVID correlates with specific IgG FC structures and interactions, for which they refer to several papers. And one of these interactions is mediated by CD16. What is CD16? Is that an FC receptor?
It is an FC receptor.
Okay. And this is produced at high levels in their fluorescent macrophages when they infect with their fluorescent virus. So when they blockade CD16 with an antibody, you get fewer infected macrophages at various time points. So I guess the bottom line is that not only ACE2, but this CD16 FC receptor is mediating virus uptake into macrophages.
Yeah,
right?
so it sort of looks like either process can happen. That paper that you guys previously discussed was really talking about that antibody-dependent part.
That's bonfire of the monocytes, right?
Yes.
That is bonfire of the monocytes.
That's Alan's. Alan snuck in and put that
I
title
slipped in and put in the title.
That's a good
really
one.
good. It's really good. All right. So, and I'm very happy they say to test whether what we see is bona fide infection. We cultured bone marrow derived macrophages and infected them with SARS-CoV-2. And they can see their fluorescent signals. They can see subgenomic RNA like the messenger RNAs. The infection is enhanced by antibodies. It's reduced by blockade of CD16, ACE2, or even RNA polymerase. I guess that's remdesivir, right? Which also reduces messenger RNA production. However, they do not find infectious virus in the cell culture supernate. I'm very glad they did that and not just looked at PCR. I was worried that they were going to just look at PCR. They say these infections were not productive. We did not detect infectious virus from these immune cells from infected mice and in vitro infected macrophages. So the virus is getting in and it's making mRNAs and protein, in the form of polymerase and fluorescent protein. Doesn't make infectious virus.
So I do have one thing that confuses me on this experiment. And maybe this is just an experience thing. When I usually think about bone marrow derived macrophages, I usually think about those being derived from mouse bone marrow. And this sort of seems like they're talking about human bone marrow derived macrophages.
I thought they were, yeah.
Yeah, and if you
Because the
actually
ACE2 blockade, right?
Right. And if you read in the methods, it's kind of unclear. And it kind of makes me think that they took the bone marrow from
The humanized mice.
And sort of used human cytokines and hoped that they got human macrophages to differentiate and not the mouse ones.
Interesting.
But they actually don't really clearly say that here.
Oh, wouldn't the reviewers have picked that up, right?
Maybe, yeah. Maybe it's just because I've only ever done mouse-derived macrophages. So I was like, oh, can you do that with human as well?
Brianne Barker, reviewer number 4.
Would you be able to get bone marrow from donors and do that? Or is it tough for people?
I personally would not, but someone would be.
Yeah, well, I don't mean you, but is that a commonly done thing? I think
don't know.
You do it in mice mostly, right?
Right.
Yeah. Okay. Point taken.
Maybe somebody had a source of human bone marrow.
Right,
that
exactly.
I don't know.
All right.
Also, I think it might be helpful to point out that probably when they submitted this paper, they had lots more explanation and
Yes.
stuff. And
Yeah,
then
sure.
when Nature accepted it, they said, okay, this is accepted, but you have to cut 50% of what's written here if you want us to publish it. And so it could be that the reviewers were privy to knowing more.
That distinction
than we
may
are.
have been stated before, and now it's not.
Right.
Could we have both versions? Could we, like, for us who want the details, could we have the unedited version?
Since you've got 15 figures online that are part of the data, it seems like you could have additional text online. Yes.
Yeah.
With another paper that I really like related to my research, there was a bioarchive version, and the full text was on bioarchive, even though the other one was in Nature or whatever. And so I actually read the bioarchive version more often. So I wonder if there's a bioarchive version of this.
That would be a good solution.
Yeah, it's a good point. Yes, and so the editors at Nature would say, Vincent, just go to the bioarchive. We don't have to do anything for you. Okay, it's fine.
But then they're referring you out to something that isn't their journal.
Yes.
Well, also it's before the peer review, and I would like to see the manuscript after peer review, which was then not cut by the Nature editors, right?
Yes.
I know I'm asking for too much. Okay, so then they say, what do the infected macrophages make? What's their transcriptome? They say these infected macrophages, human macrophages, preferentially produce a chemokine called CXCL10, which recruits other immune cells, but they don't overproduce tumor necrosis factor. Now, to me, that doesn't mean anything, Brianne. What does that mean to you? No TNF, but high CXCL10. Does that mean anything?
Not particularly.
Okay. So it's not a signature. It's not an inflammasome signature.
No, it's not something that really jumped
fine.
out to me.
Good.
It might be helpful if anybody is trying to follow along with the manuscript to say that we're up at the beginning of figure 3 now.
Figure 3, yeah.
It's around line 144 in the accepted article preview.
And so they say the production of this chemokine is enhanced by antibodies and inhibited by remdesivir. I guess the anti-spike antibodies that allow entry into the macrophages. They should specify which antibodies. They shouldn't just say antibodies there, right?
Yeah.
Inhibited by remdesivir. So they say we use this chemokine as a proxy for viral infected macrophages. And they said then we determined the transcriptional signature for these tissue-resident macrophages, the lung we're looking at here, after infection. They say in particular alveolar macrophages. So they have a variety of transcriptional signatures for alveolar macrophages, intestinal macrophages, inflammatory cytokines, complement genes, and so forth. But we don't need to go through the details there. And then they say if you look in the lungs of people who have died of severe COVID, they have distinct transcriptomes, alveolar macrophages from those patients. They have transcriptomes that mainly are in line with what they are seeing in the mice. that fair enough, Brianne?
Yes.
Say that? Okay. I like that they are doing that. They're comparing the mouse data with the human data. Appreciate that. All right, now we get to the inflammasome. And they say, if you look at these fluorescent cells in the lung, they have membrane bubbles, which are characteristic of pyroptosis. You know, remember the old saying, ruffles have ridges? Pyroptosis have membrane bubbles. So they said, we therefore looked at inflammasome, the inflammasome as part of this, whatever's being activated. What is this membrane bubble business, Brianne?
So when we think about apoptosis, apoptosis is a form of cell death that is characterized by the cell making little sort of blebs. Little compartments where it's sort of taking all of the intracellular components and putting them in little packages as the cell breaks down.
Okay.
And I like to tell my students that it's very quiet cell death. It's when the cell is dying and doesn't want to disturb its neighbors. And
it's just packing up its stuff and sending it out.
Exactly.
Yeah.
And pyroptosis is different in terms of there is no packing stuff. There is no kind of organized process with the membrane. Here, we're just basically getting what's really a programmed necrosis or a programmed lysis of the cell. And so this one I think of as loud cell death.
Burn it all down.
Where the cell is dying and it is also warning its neighbors that they may need to be in, say, antiviral state, or they may need to turn on a defensive program. And so the membrane bubbles are really kind of partially an indication that it's not apoptosis.
Okay.
And starting to make you think about pyroptosis.
All right, got it. They actually put a sentence in here that describes what an inflammasome is. Inflammasomes are dynamic multi-protein complexes in which specific NOD-like receptors and adapter molecules are assembled to activate caspases, the central effector proteins. So they basically sorted out cells that were fluorescent, either positive or negative, human immune cells, epithelial cells, and mouse immune cells. And they say assayed for sensors, adapters, and effectors of the inflammasome pathway, which is probably a sentence that is like multiple panels.
Oh, yeah.
And they focus on ASC, this adapter molecule, apoptosis-associated speck-like protein containing a card domain, ASC. This has a pivotal role in the activation and assembly of the inflammasome. So that you look for ASC speck formation, right? That's a
So basically,
assay,
what happens is that this protein is usually broadly distributed throughout the cytoplasm, and if the inflammasome is activated, then multiple ASC proteins come together, where this multi-protein complex is forming, and you get this little speck in the cell that you can see pretty easily.
So they use speck formation as kind of a quantification of inflammasome activation then, right?
Yep.
Okay.
And the nice thing about using ASC is that it doesn't really matter which sensor you are working with. You pretty much always need ASC. So that gets you around figuring out which sensor is involved.
Cool. Very good. All right. So what they find is that the infected fluorescent human cells exclusively show inflammasome activation as measured by ASC speck formation. These specks co-localize with activated caspase 1 and NLRP3.
All right, so if anybody's following along and you're looking at figure 3F,
right,
it's the cells that have the yellow spots, the speckles. Those are the ones that have the ASC and they're the infected ones have those.
And they say that this activation that they're measuring is sustained during disease in this mouse model. Okay. Now, once you form these inflammasomes, you make, as we said, activated caspase 1, which then activates the pro-inflammatory IL-1 family of cytokines, like IL-1 beta and IL-18. And these are, as we said before at the beginning, these are typically elevated in severe COVID patients. They find in their infected mice high IL-18 levels in the blood and in the lungs, and this correlates with the numbers of fluorescent macrophages that they see. And they notice that we don't see high IL-1 beta, but we do see high levels of this IL-1 RA, right, which is the antagonist, right? So that's kind of neutralizing the IL-1 beta. Is that right?
So it is.
It's a decoy, right?
It's a decoy, and if people want to hear more about that, they could listen to TWiV 882.
That was you and I doing that, right?
Yes,
Yeah.
Where we talked about a paper specifically about IL-1 RA.
Oh, yes.
And vaccines, and that paper is so cool.
The two-component activation in
Yes,
the mRNA vaccines. Yeah, that was cool. That's cool. And then apparently another assay for pyroptosis is by LDH, which we mentioned, and Gasdermin D.
Yes. So Gasdermin D is actually the pore that opens the membrane during pyroptosis.
So this is produced as part of the activation, and it makes pores, which is part of killing the cell.
Yes,
right?
exactly. So it makes the membrane pore that leads to the cell death.
And then the pore allows LDH out, right?
Mm-hmm.
Okay. And I find that LDH and Gasdermin D are elevated in the serum of mice at late time points. And here we have our zombie aqua again. A small fixable dye that enters dying cells. Does it enter through these pores pretty much?
That's the idea, yeah.
That's the idea.
So,
I mean,
Leanne,
any compromised membrane is where it will go in, but the pores are a pretty good place where the membrane is compromised.
are these cells being killed, or are they committing suicide?
Committing suicide.
Suicide.
That's what I thought.
So, Brian, if a CTL were killing a cell, would zombie aqua get into the cell as well?
Zombie aqua could get into the cell, though that would be apoptotic.
Yeah. Okay.
Death.
But you said any compromised membrane.
Yeah, it would.
Yeah.
It would be about. It would matter sort of how far into the apoptosis you were.
So I guess any virus that encodes a pore and compromises the membrane, you might be able to sense that by zombie aqua.
Right.
Okay. Anyway, so they see higher incorporation of zombie aqua into these infected lung macrophages. And so, but they say is all aspects of inflammasome activation are also, all of these things that we've talked about also happen when you infect bone marrow-derived monocytes from in vitro with SARS-CoV-2. So everything we've talked about so far is in mice. All right. Next. Let's see. This is called infection impacts macrophage response. So what's the role of infection on the inflammatory response of these macrophages? So if you block entry with antibodies to ACE2 or CD16, if you inhibit replication with remdesivir, you reduce all of these things, IL-18, IL-1RA, CXCL10, and those all parallel decreases in the fluorescence that you would see. If you deplete CD16 positive cells, you don't see any increase of IL-18 and IL-1RA. So they say this is consistent with the idea that most of this is happening in myeloid cells. So is that a myeloid-specific marker, Brianne?
It is mostly on myeloid cells.
Okay. Antibodies that promote infection. Remember, we said certain spike antibodies promote infection of macrophages. They enhance IL-18, IL-1RA in the blood and CXCL10. But none of these monoclonals or CD16 blockade make an impact on lung pathology. And they say that's probably because, you have to balance viral replication with inflammation. If you knock down inflammation, you still have viral replication, which is damaging the lung tissue.
Right.
All right. We're almost done, folks.
Yeah,
I know it's a lot.
They actually have a really nice place at the very end of the abstract where they explain that point really well.
Yeah.
where they say that the same blockade of inflammasome activation leads to release of infectious virus by the infected macrophage. Thus, inflammasomes oppose host infection by SARS-CoV-2 by production of inflammatory cytokines and suicide by pyroptosis to prevent a productive viral life cycle.
Right.
I had a really hard time with that sentence. I probably looked at it for about 20 minutes because I decided that the inflammasomes, it just says anyway, the way you read it makes a little bit more sense, but this is the place where I thought they were missing a verb or a verb form.
It's kind of like reading Shakespeare, you know. You have to have the right sort of inflection, little body language, the whole thing.
Yeah.
Well, after 10 minutes, I say I'll let the other people on TWiV figure it out.
Well, good thing Brienne's here because I'm not one of the other people who can figure it out.
Sometimes other people do too. Yeah, sometimes others, but yes, for the paper like this, we wouldn't do it without Brienne present. No. We'd be wandering all over the place, and then she'd be screaming at the computer.
We'd be Googling immune cells left and right.
So, so we've talked about caspase activation, inflammasome activation. What does this have to do with disease? So they treat mice with caspase and NLRP1 inhibitors.
Now we're on figure 4.
Figure 4. And so, again, the proportion of infected cells remains the same, but the inflammatory profile changes. It's attenuated. It's reduced, right? Because you're inhibiting. That's the idea, that you want to know if the inflammasome activation is causing all of this, and it looks like it does. With these inhibitors, the mice make less chemokine CXCL10 and also less in the serum. Overall, the treated mice have lower levels of caspase activation, IL-18, IL-1RA, and gasdermin D. And they say sorry, go ahead.
No, go ahead.
The cumulative decrease of all of this reverses the immune pathological state of the lung by looking at lung histopathology. So you inhibit inflammation and you reduce the pathology. And it also reduces immune cell infiltration, which you would expect.
And it's a specific way to inhibit inflammation, right? We're targeting the inflammasome.
Right. And this is interesting. It reduces immune cell infiltration, right? Because you're knocking down this CXCL10, which recruits a lot of immune cells, enhances tissue recovery, but you still have a lot of infected cells. You're not really doing anything to impact that. And these are all reproduced in cultured cells in vitro as well.
Well, it's not just that you still have a lot of infected cells. You actually have more infected cells because you're not seeing the cell death of those infected cells. The infected cells are not actually dying to sort of save everybody else and stop being viral production factories.
I was just going to make a comment in case they have a chance to fix this at the proof stage, which maybe they don't, but where they say infected cells did not diminish, that's actually figure 4B. Mm-hmm. And then they say, but the inflammatory profile was drastically attenuated. And there they reference 4B, but it's actually 4C. So again, minor point, but for somebody like me who's thinking, oh, I can probably look at that figure and actually see if I believe what they're saying. And then I look at the figure and it's like misdirecting me. So, yeah.
Finally, last experiment. Does inflammasome activation have any impact on infectious virus? So they treat mice with caspase-1 inhibitor. They infect them. They measure virus in the lungs. And these mice have higher viral loads 14 days after infection. So they say, that's interesting. Maybe the reduced inflammation could make more virus, right? So they infect macrophages in culture, treat them with the caspase or NLRP3 inhibitor. They make more virus. Remember, a couple of minutes ago, the macrophages that are infected don't seem to make very much infectious virus, but now they do when you inhibit caspase-1 or NLRP3. And I read this and I'm like, wait a minute, they just said, but then I realized that they're treating them with this inhibitor. So they conclude activation of the inflammasome plays two functions. It reduces virus production and then it signals the immune system, by releasing these chemokines and so forth to get more cells in to help things.
So
Very
we're
interesting.
talking about this macrophage response and this immune response that's pathological and causing COVID-19, and yet what's happening is the macrophages are really just doing their job. They're
doing their
this
job.
is the response that you'd expect the macrophages to have, but it causes a problem.
But it's only in 10 to 20% of the patients that you have this issue, and that's the question which we don't have an answer to. Why? What's the? I mean, I think they say in the discussion that if you have a poor initial interferon response, this is probably leading to this, right, Brianne?
Sure. I think that could make a lot of sense because if you have a poor initial interferon response, you're probably going to make more virus, and then the macrophages are going to have to do more to try to control that virus.
And then it gets out of control. So do we know if people with severe COVID have SNPs and interferon genes? Has that been looked at? They do.
Or at least in interferon receptor and related genes. I think that Casanova's lab did that a while ago.
So there must be a little of this stuff going on, even in a well-controlled acute infection. But if it's not well controlled, then this gets overwhelmed.
Right.
Okay.
So a very nice summary. These findings suggest infection of macrophages and activates the inflammasome, drives pyroptosis, which interrupts viral replication. It releases immune cell activators and recruiters, and the cytokines as well as viral RNA released from cells likely shape the hyperinflammatory macrophage response and drive immunopathology.
So I actually was going to cite the last sentence of the previous paragraph as being even better than that sentence I couldn't get in the abstract, where they say, the activation of inflammasomes in infected macrophages plays two protective functions. It attenuates virus production and signals infection to the immune system by releasing inflammatory cues to recruit and activate more immune cells. That, to me, is the best summary of the whole paper. But
in fact, the last paragraph, they say, a role for inflammasome-driven hyperinflammation in COVID pathophysiology is now recognized in patients. Targeting inflammasome pathways may provide alternative therapeutic options. However, increased virus production seen upon inflammasome blockade could be a risk.
Yes.
Right? You have to balance the two.
So maybe you treat with a combination therapy at that point and block inflammasome and also treat with antivirals at the same time.
Yeah, and the useful thing here is, if you remember back at the beginning, we talked about dexamethasone and how dexamethasone is an important drug that's used for treatment, but it's pretty broadly immunosuppressive.
Yes.
And so here we could think of a particular target to more specifically target just the pathologic immune response and allow protective immune responses to still keep happening, like the CD8 T cell response.
Right.
The key is timing, because you don't know who's going to have severe COVID until they do, right? So you have to have a patient coming to the hospital who is low oxygen, right? And then you have to decide where they are. And at that point, probably an antiviral is not going to be helpful, I think.
At least not in isolation.
But maybe together with these inflammasome. And we'll see where this goes. Very
next time around.
Yeah.
Maybe we'll be able to just look at a person's genome sequence when they come in.
That could
and
be.
know whether or not they're predisposed by some, interferon SNPs or something like that.
One would hope, right?
To severe disease.
Well, of course,
the profile will be different next time around.
Yes, it will be. Yeah.
And it's likely not just one thing, right? It's going to be.
Yeah.
But it's a good argument for having everyone's genome sequenced, right? So then the doctor looks at their chart and says, yeah, you have these SNPs, we're going to give you this, right?
If we can do that without horrific consequences, yes.
Yes. Well,
which I have very low faith that the U.S. medical system is capable of.
The implication is that by the time we're able to do that, which might be decades from now or even a century from now, that's when we'll be in the next situation of needing to quickly identify people susceptible to the new virus.
Sadly, we won't be around to talk about it.
Right.
Oh, wow.
But if it doesn't happen until we're gone, I'll be happy.
Yeah.
Yes, right. If we don't have to deal with this again in our lifetimes.
A lot of good stuff. But let's do some picks. Brienne, what do you have for us this week?
I have a big science story that actually came out earlier today. There was a multi-group press conference from multiple universities simulcast at the same time that they have taken a photo of the Milky Way's black hole. So the black hole at the center of our galaxy, and show some photos of that black hole. So this is sort of the second black hole to have been photographed. It looks like a donut, as expected and as it was predicted to. This one is much smaller than the previous, and also moves much faster. So it was a much larger technical hurdle to photograph, but it is sort of in our neighborhood. It's called Sagittarius A star. And that's kind of what makes up the Milky Way.
Yeah, that's the like the sun of the Milky Way. It's the
Yeah.
And we're that much
Cool.
closer to finding all the lost socks.
How far away is this?
Let me see. That I don't know.
All right, don't worry
I
about it.
forgot what the distance was on that.
It's pretty far, I guess. Neat.
I'm pretty sure the answer is pretty far.
Yeah.
And what did they use to take this picture?
so
An
they-
iPhone?
No, so it's actually a collaboration with multiple telescopes. So it's 300
Okay.
scientists, 13 institutions, and I think this one had 8 telescopes
Wow,
at the same time.
cool. Very cool. Kathy, what do you have for us?
I have something that inadvertently kind of relates to that first TWiV when I was on as a regular host, which wasn't the
Yes.
first TWiV I was on. But this is a story about river dolphins playing with an anaconda and capturing it. And first of all, I didn't know that there was such a thing as river dolphins.
Yeah.
So that was maybe the main take-home message for me. But there's a photograph and then a description of these river dolphins that kind of were teasing this snake. And they think that the snake did not survive, but there's no evidence of the river dolphins eating it per se.
They're just playing with it?
Yeah.
Like
from-
cats
I
with
mean,
yarn. Yes.
Yeah.
Poor snake.
Bolivia.
Bolivia.
Yeah.
There are river dolphins, although there are fewer and fewer, and there may not be for much longer, is my impression.
I think I had read something that they thought it was sort of like showing the kids showing the juvenile dolphins how to play or something like that. It was.
Could be.
I
Or it
really
could
liked that image.
Yeah, it could just be for fun. It could just be, hey, look.
Tell you, I wouldn't want to play with an anaconda. That's,
No.
No, these dolphins have sharp teeth, looks
Yeah.
like, and
Yeah.
there's 2 of them ganging up. There's no chance
Yeah.
for that snake, you know?
Yeah,
It
and
could be-
anacondas, I don't think they're venomous, are they? They kill by
No, they
constricting.
squeeze you.
Yeah. So they're not, this snake does not have a chance against these 2 dolphins.
Too bad.
Well, I mean,
That's nature,
just
man. I know.
yeah, that's
I know.
nature. And we've got more anacondas than we have dolphins. So,
So these are freshwater dolphins, is that right?
Mm-hmm.
Wow, it's interesting. I'd never heard of that.
yeah.
Yeah.
How cool. It's
I think
in the
that
Journal
there are
of
some
Ecology.
in addition to the ones in South America, I think there might be some in China, or there were, or I remember these are definitely endangered.
Cool. Rich, what do you have for us?
In honor of Smallpox Vaccination Day, which is day after tomorrow, probably about the time this drops, September 14th, which is the date in the year when Edward Jenner inoculated James Phipps in the first deliberately experimental smallpox vaccination, vaccinated him with cowpox. In honor of that event, I have an article called Smallpox Vaccination Techniques from Knives and Forks to Needles and Pins by Derek Baxby, which I believe is published in 2005. Unfortunately, this is behind a paywall. I even wrote the journal Vaccine, and asked them if they could make this open access, at least for a time, in honor of Smallpox Vaccination Day. But it hasn't been done. At any rate.
Do you have access, Rich?
I do have access.
Okay. Looks like I do too. I was going to send it to you if you didn't.
No, I got it. Because I will be totally honest with you. I haven't read this thing. Okay. I just thought it was cool. And the pictures are cool because it's clearly a history of all the different tools and techniques that have been used in smallpox vaccination. And I should read it because I think that the actual techniques that you use impact the response that you get. Derek Baxby has written extensively on all things orthopox virus and smallpox. He's got a couple of histories of the eradication campaign and the disease and everything else. And this is specifically about vaccination. So I thought people might be interested in that.
It's very cool.
Look at those
I do
pictures.
not have access.
The pictures are great.
I'll send it to you, Alan,
Okay, thanks.
So,
and anyone else.
Rich, the kid that he inoculated, Phipps, Phipps, right?
James Phipps.
No. So what, did his parents volunteer him to do this or what? How did that work?
Actually, I don't know. I don't know. But of course, this is in the context of he was probably at an age where he would have been appropriate for inoculation anyway.
Yeah.
Okay. So are you going to inoculate this kid with smallpox and take that risk, or do you want to try this clinical trial?
Yeah.
I'm just curious about how it went. That's all.
I don't know the details of that.
Yeah, because I know it's not a time of medical consent or anything
It may
like
have
that.
just been, I'm going to do this. Yeah.
But Phipps maybe said, I want to do it, or maybe, how would he know? How would he understand?
Yeah, he's only 8 years old.
I think
probably.
Jenner may have just decided to.
Or his parents may
Or
have.
parents may have.
I suspect it was the parents, yeah.
For some reason, my understanding of the story was that the second person that Jenner did was his own son. So
That's interesting. Well, Albert Sabin, before doing clinical trial of OPV, he gave it to himself, his wife, and his 2 daughters.
Yep. This is a long tradition.
But they're all seropositive, so apparently had been infected, right? Or maybe they'd gotten IPV, I don't know. But so, yes, that's a long tradition which doesn't happen any longer.
Maurice Hilleman, one of his daughters, got one of his vaccines.
Yeah, there was a guy from Russia who discovered hepatitis E virus, and he drank some filtered diarrhea to prove it. I mean, and I saw him at a meeting, and someone said, "That's the guy who drank the diarrhea." My gosh, he was walking around with a toothpick. I mean, who has just toothpicks walking around anymore? That's what happens when you drink diarrhea. Alan, what do you have for us?
I have something that actually connects to my previous pick, which was a museum of old radios, and then I was reminded of this link that I've had in my queue for quite a while. This is a video put together by AT&T in 1940 about tremendous one of the greatest inventions of the 20th century, the vacuum tube. And everybody says, "Oh, now vacuum tubes, those have been replaced." Well, they got replaced with transistors, which came from wanting to develop a new type of vacuum tube, basically. Yeah. And so this video goes through. I picked it for a few reasons. First of all, it's just kind of cool, and it's got the traditional, the narration style of 1940.
Well, it's got Lowell Thomas,
yes, right?
Yeah.
And also because it has one of the best explanations of the electrical mechanism behind a vacuum tube that I have ever seen. So it's a great explanatory video from that perspective. And because a vacuum tube does the same thing that a transistor does, this is the concept you need to understand modern electronics. Like, what exactly do these things do, and why is that important? So this is the key to amplifying electrical signals. And then you take transistors and you put a billion of them in one place, and you've got a computer chip. Well, not a billion of them necessarily, but the whole concept of computing is based on this. Anyway, there's also the fact that this is before World War II, but you'll notice something about the people working in the vacuum tube factory that's just kind of interesting. They almost all of them are female.
Yeah. Yep.
So these tubes are being built entirely by women in this factory,
Yeah.
Which is still who builds electronics, because women on average have better fine motor skills. And so
It's interesting.
cool.
My course this year was in a chemistry building. Yeah, Havemeyer Hall.
Mhm.
And outside the lecture hall, where I always waited for 5 or 10 minutes, so there's this display case about Alex Langmuir,
Yes.
who figured out how to improve the light bulb by putting something else in it besides just evacuating it, right? Something
Right.
like that. And they have pictures of the manufacturing. They're all women.
Yep.
And they have long, nice-looking dresses on. I mean, they're manufacturing stuff, and they're nicely dressed.
They're nicely
you know?
dressed, because that's what you did in that time
Very
period.
interesting. And then they have, of course, models of all the light bulbs. Some of them are really scary. They look like I would never screw one in. It looks like it would explode. Cool. Oh, by the way, you can still buy things with vacuum tubes in them. Then some audiophiles appreciate that it's a different sound, right?
I have
we have
one of my ham radios actually uses
Has vacuum
tubes in the final amplifier.
So
It's an
there
old
are.
radio.
I have these pieces of equipment called channel strips. They're basically, you put your microphone through it and it amplifies it and does some other things. And you can get them with vacuum tubes, and often recording studios will have a selection of vacuum and solid state just to get different sounds. And there are even ones that have both, so you can switch in between, you can blend, and they give it a unique sound that you can't get by solid state.
Well,
quite
the
interesting.
world is analog. We just choose to represent it digitally. So if you
Indeed.
use an analog device like a vacuum tube to amplify, or even analog transistors,
Yeah.
you can get a different effect.
All right, so my pick is a set of books by Pamela Jane. So Pamela Jane is a listener. She emailed me a while ago, and she said, I write kids' books. Would you like to give some of them away, have a contest? I said, sure. So she sent a case of them. I'm going to show you some of them. But she wrote in a letter, I'm enclosing a selection of my children's books to give away to your listeners with young children. I hope they enjoy them. Coincidentally, when I was looking for something else just now, my dad, Dr. Philip Stanley's patent for preparation of polymyxin popped up on my computer. I'm closing the first page just for fun. So this is, apparently her father got the patent for polymyxin. This is 1947.
Wow.
I tell kids at school visits that he got only $1 for his work, but he never complained. Like you, he did what he loved most. It appears I didn't inherit the science gene, but my dad was also an accomplished writer, so I got some of that fairy dust. Thanks for all you do. Your TWiV listeners are very grateful. So we're gonna have a little contest to give away these books. There are a bunch of them. So we have here Halloween hide and seek. We have Noel and the Nutcracker. These are not science-based, okay? These are just kids' books. I have a couple of copies of that. Then we have, this one is for very young kids. This is Spring is Here. It's got, pop-up stuff.
Cool.
I've got 3 of those. Then we have this one, which I have 4 of. A vampire is coming to dinner. A vampire has invited himself over to dinner. Interesting. Then we have, I jumped over 3 pots in a pan.
C jumped over 3 pots in a pan.
C, yeah. And landed smack in the garbage can. I have a couple of copies of that. I have Monster Mischief. They're all nicely illustrated, as you would expect. I have the little elite one.
Elfie.
Sorry. Elfie, you're right. And then
Vincent is going to use these to learn how to read.
Little Goblins 10. Well, I'm kind of reading them upside down.
Yes, I know.
And so anyway, I would like you, if anybody wants these, I'll send them to you, but write a little verse, write a COVID verse. And I really loved our COVID verse contest. It doesn't have to be long. And in fact, if you wrote a verse and you didn't get a virology textbook, you could resend it and you'll likely get one of these, because I have like a dozen or 15 or something of those. Okay, what's the email? TWiV@microbe.tv. And I don't think there's going to be a big demand. I don't know how many people have kids, so.
Did she send you Pride and Prejudice and Kitties?
No. No, that was all
man.
she sent. The other thing she
Okay.
sent was so it's kind of an autobiography of herself, which I'm going to read.
Yeah, she's got a memoir.
I'm going to keep it. It's autographed.
Nice.
I will give that away eventually, but I want to read it. She's the author of over 25 children's books and Pride and Prejudice and Kitties, a cat lover's romp through Jane Austen's classic. That's cool. She's a writing coach. Yeah, there's a picture of her in the back there with her kitty. Thank you, Jane. Pamela Jane over in Pennsylvania. And that will do it for TWiV number 900. You can find the show notes at microbe.tv/TWiV. You can send your questions and comments, your verse, COVID verse. You know, I was reading some of those the other day. Some of them they're just fabulous.
Yeah.
Jabberwocky one, and oh my gosh. TWiV at microbe.tv. And if you like what we do, consider supporting us. You can go to microbe.tv/contribute. A couple of ways you can do that, and they're all tax deductible, federal U.S. tax deductible, because we're a nonprofit entity. Alan Dove is at alandove.com. He is Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Thank you for 890
890 I've been on.
roughly, give or take.
I missed a lot of them, but
And Kathy mentioned that wasn't the first time. I think all of you were on another episode first to see what it was all about.
I was on as a guest. Actually, episode 10, I was on as a guest.
But then you came on the very
And then you
next
couldn't
one.
get rid of me. Yeah.
And then so Kathy
kept coming
came on
back.
earlier as well. Rich Condit's an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun. And it's been a lot of fun to listen to 900. It's actually more than 900 because you had some episodes that you didn't number. They were
Yes.
special episodes. Yeah.
Special episodes. Yeah, it's not really fair. I should have numbered them all, right?
It's all right.
Well, it's done. Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.